here, and let's show you how we map all the different genes that are on this array. So I'm going to open up one of her arrays. Okay, so here's an array, and now I'm going to map it so that in this map, just like a road map or anything else, it tells us what street we're on or what building we're looking at or whatever address, and that address tells us what gene happens to be there, or at least the sequence of the gene. Some genes, nobody knows what their function is, obviously, but at least with the sequence, maybe in time we can run it through some bioinformatics and figure out what that gene function is. Anyway, so here's a really beautiful array, and you can see that it has on the corners, let me just increase the brightness a little bit. On the corners it has little placements, and this is where it tells us where to put them out. So it's like telling us north and south and things like that. So those aren't actually, these 5 spots here are actually, again, just to help us place our map onto this array. And so we call maps, in the case of these programs, gal files, and so I'm going to load an array list. And so I have this one that says that I put together from an Excel kind of file that has all the genes. This is our gal file. And so here's the gal file. Notice it actually shows up for 4 arrays, but we're only going to work with one, so I'm just going to take the top one, because each one of these would be called a block, and I'd rather just use block 1 for everything. And I'm just going to drag it down the map to our actual array. Now I'm going to zoom in, and I'm just going to make sure, now obviously there's a lot of circles there, and I'm going to, whoops, I'm moving the map around. There's a lot of circles there, and it's easy if you moved them in the right, slightly off, these 44,000 spots, you could end up having kind of like a frame shift error. So you want to make sure that this map is on the right spot. So we're going to go over to, I'm going to see, so here, let's just show you, see there's the array, there's the map. I'm going to move those circles right over the top. I'm going to zoom in to make sure I got it. See, so I'm just going to make sure that these 5 fit right there. See that? Now I'm going to zoom over to the other corner, and you can see this, they're almost lined up, but they're just a little bit over. But you want them to be nearly on top of each other, otherwise, because each one of these circles represents an address for a gene of interest or potential gene. Oh, so if you notice the map is just a little bit off. So I'm going to drag this corner a little bit until it fits these 3 place markers here. Now I'm going to go down to this corner. And then you see, oh, there's another place marker here. I'm just going to kind of make sure that it's kind of on it. And then I like to just double check the corners. You can now see that almost everything is circled. So now that each circle will be, the computer will tell you the intensity of that circle. So I'm going to take it on over. I just like I said, I like to double check corners. So this one's just a little bit off. It's down just a little bit. Now what I like to go check the corner again, right? Because I might have moved it a little bit odd. So you can just kind of keep moving it around a little bit until it's what you think is perfect. But here's the so this isn't too bad now. See how it's mostly lined up? So I really feel pretty comfortable about this. Now I can tell the computer to find the features. So now it's going to join the map with the images, because the computer can pick up the pixel intensities. These are fluorescent units, these intensities, also known as FU units. And so the computer will pick up that, and those signal intensities would then be statistically analyzed in another software program. So let's go ahead and hit this. There's a hotkey is what they call it, where I'm going to tell the computer to find these features. So I'm going to push F5. And now it's going to go through and look at the image. It's going to look at the block, and it's going to find all the features in that block. And again, these features are also called spots when I've been referring to them. Now you notice good looking spots have clean circles around them. And the circles kind of increased in size and decreased in size to take in the entire area of the spot, because not every spot is a perfect size. Now you notice there's some spots that have a line through them. That's flagged data. So that's data that's by the computer considered to be non-existent. And you can actually go through, if you wanted to get real crazy about it, and flag even more data. I'm going to pick the right button for that. So I could actually flag that data and call it bad. See how I put a cross through it? Or let's say I went through and I said, oh, wait a minute, this is really a good one. This should be considered. I could go back through and say flag good. So you can do all sorts of things like that. My opinion is the statistics will usually clean this all out. Even if it flags something that it shouldn't have been flagged or whatever, I'll have the signal intensities, and if it's consistent, it'll be statistically significant. If it's not consistent, then it's going to be washed away anyway. This is why, and so having perfectly nice chips makes the statistics very easy to do. And sometimes you might want to throw out an array that doesn't look as good, just because it's going to ruin the statistical analysis. But when you have a beautiful chip like this, that's the way to go. So now I'm going to hit this Analyze button here, and it's going to go through and read all these circles. Remember, there's over 44,000 circles there. And it's going to give me the intensity values for all of these circles, and then I can take that back and figure out what those genes are, and do statistics on those genes. Okay, so now it's showing you, over here it's showing you different gene names, and if there's flags. So look at this one here. This is showing you that this one is, so this is going back to the original map, and it's showing you that this gene was turned on in the green treatment, but not in the red treatment, whatever that happens to be, whether it's a bacteria or whatever. Actually, they're both different types of bacteria, but this gene is being turned on. And then you can do statistics on it. It tells you, it keeps track of all the different intensities. It tells you the signal value of 635, because that's the dye that was read at. It gives you the signal for 5 information. It tells you whether it's flagged or not. And then it all can be uploaded into a statistical program. Here's the scatter. So the histogram showing that the two images are nearly 1-to-1 ratio of relative amounts of yellow to red, and the statistical programs or whatever can actually bring these numbers a little bit closer together mathematically. So one's a little bit brighter red, it might bring the red down a little bit and bring the green up. It usually does it on a log scale to help smooth the data. And then we do this over all the different, and we'll scale all these other arrays together. So anyway, that's the gist of how to scan and map out your arrays. And then in the next video, we'll talk about how to statistically analyze the data. Anyway, thanks for watching, and we'll have another video eventually on this.